the last slide of this PowerPoint that I have posted for you guys, but I purposely blocked out, right, each one of the things. And remember I said last time that drawing sometimes helps people. And so this one is, it's not very detailed drawing, right? We're not talking about really, artistic drawings here that can sometimes really help out some of us that are real visual type learners. Can you guys get that door for me? There's somebody noisy out there. So we talked about clonal selection last time and going through that process all the way from first exposure to second exposure. So you remember first exposure is the very first time a B cell or a T cell sees its specific antigen, right? That very first primary first exposure. Remember B cells develop where in your body? In your bone marrow, and they leave there educated, they have their receptors on their surface. So although this doesn't look like it, but what is another name we give to the B cell receptor? It's another name to the B cell receptor. What is actually coating their surface? It actually has two names. Immunoglobulins, so what's another name for an immunoglobulin? Antibodies. Right? And remember there's 5 different classes of antibodies. There's IgM, E, A, G, and D. Which one is on the surface of your B cells? M. Good. IgM. In this monomer form. Now, what thing I don't like about this drawing, and we'll fix that on Friday, is that it doesn't look like an antibody because what is it kind of missing? What do we usually associate, what shape do we kind of associate with an antibody? The Y-shaped. And this is kind of a Y-shaped, but it's not in enough detail that we have 2 antigen binding sites on either side of this, right? We would have 2 antigen binding sites. Instead of here, it's really only showing 1. Do you see how it's connecting with just 1? Right? So if they hadn't labeled this a B cell, I would have said, oh, this might be a T cell. But the other reason why I know it's probably a B cell is that this is kind of an antigen all by itself, right? And one of the major differences between B cell and T cell interactions is that B cells can directly interact with an antigen. Say this is a virus, a bacteria, right? Some foreign protein, some toxin. B cells can interact directly with it as you see here. T cells, can they do that? No. They have to have the antigen presented to them by one of our cells, right? By one of our cells. And we'll look at that interaction more closely again today. And we're going to draw all this stuff out on Friday. That's why I said I'm going to actually get up and use these boards that are in this classroom, right, and work on it together. So B cells, right, can interact directly with the antigen. And remember, this is at a specific little tiny piece of the antigen. What do we call that little tiny piece? His little crown you see here in this picture. No, the piece of the antigen that the antibody is binding to or a receptor is binding to. Antigen binding sites are the sections on an antibody. But antigens, they're really big, but where the receptor or the antibody is actually binding, we call an epitope or an antigenic determinant, right? And I'll always say epitope. Right, so that little red thing is the epitope, right, of this antigen. They say it's a bacteria, right? Scary little bacteria. Okay, so that's first step, right? The very first time B cell sees antigen, it binds to it. Is that it? Is that the end of the story? No, right? What's going to happen next? What's going to happen next? So we have this one B cell all by itself. It's recognizing antigen. Can anyone tell me what's going to happen next? I'd say we're going to stay with just one B cell. We're going to select just one B cell. He's going to have to do all the work all by himself? No, right? we want to select him to proliferate, right? Now, can he do this all by himself? No. Sometimes he needs who? Who's going to help? The helper T cell, right? So here, again, not showing that interaction that we're going to draw in more detail on Friday, but they literally going to join up with each other, right? And by receptors, right, recognize each other and send signals. Now, this is if this antigen is what we call T-dependent, that we need this T helper cell, right? There is a shortcut for some antigens, right? String response, where they can bind to lots of antibodies on the surface, like lipopolysaccharide, and we can skip to step 3 right here, right? Where we get the proliferation. But does a B cell stay a B cell? Do we just make a whole bunch of B cells? No. Some of them are going to turn into what cells? They actually secrete antibodies. What's their name? Their rough endoplasmic reticulum gets really big. Plasma cells, right? And then my favorite group of cells, right? Second none to, of course, the natural killers. But the great thing about our adaptive immune response is that we produce those plasma cells, but what other set of cells are going to be long-lived and remember this guy when he comes around again? Memory cells, right? So those are our 2 important subsets, right? And we're going to make thousands of these, right? B cells that are going to differentiate into plasma cells. There's a reason why he has a little mouth here. He's going to spit them out, right? And the memory cells, right? They're going to have those receptors on their surface, and they're going to hang out in your body in case we see this guy again. So we are not going to have to wait to find this one lone B cell. We're definitely going to have quite a few B cells that recognize this specific bacteria at this specific epitope, right down to that molecular level. So remember, how long does it take to go from this B cell recognizing this helper cell may be helping out, or skipping right to this phase where we have plasma cells and then we're secreting antibodies? 7 to 14 days. On average. That's a significant amount of time. Remember, I was really mad at my TV show Scorpion that I didn't even watch this week because I was sleeping. So I can't even complain if they screwed anything up, right? Somehow, miraculously, this happened in like hours. That show. No. Days. Okay. So now they're showing you here antibodies can bind directly to that antigen. Now we know that that's going to cause the immune system to do something, right? If you have phagocytic cells and they see that bacteria coated in antibody, what are they going to do? If it's a phagocytic cell, they're going to engulf it, right, and eat it up. Okay? And then think about those other protective outcomes, right, that we're going to go over again on Friday. We're going to write all these out, right? That cross-linking that happens. If it's IgG or IgM, it can activate complement, right? If it's IgG, if a natural killer cell comes along, we can particularly kill that cell. If it's a virus-infected cell. Right, so we can expand upon this discussion here. But let's move forward, okay? We clean up all this mess, right? And this is one of the examples, right, of how we clean it up, is that notice that the antibodies are bound to the bacteria on its epitope, and the phagocytic cell, I think this one is actually a dendritic cell, the cartoon for the dendritic cell, has receptors on its surface, and it's going to gobble them up until we get rid of them all. This next step you could almost put in a box by itself, right? Because when does a memory cell come into play? this should be the end of the story, right? For the very first primary response. We've eliminated the invader, we've survived, we lived. This is, when does a memory cell come into play? The second time you see something, right? This is the second time, or the third time, or the fourth time. Any subsequent exposure after the first, it's probably going to be a memory cell. And do we go through this process all over again?
No.
Yeah, we do. We do. Each time we're going to maybe have the T cell, but in this case it'll help out the memory cell, right? It's going to differentiate into plasma cells and even more memory cells, right? It's going to secrete antibodies. Those antibodies are going to help us eliminate the invader. And then if we see it again, the cycle starts all over again, right? Every time. The only difference is that this time, right, from memory cell to antibodies is a much shorter time frame. We're not talking about days to weeks.
So the second time, it doesn't make as many memory cells?
No, the next time it's going to make probably just as many memory cells. So that's why, too, we spoke about this before, especially people who experience allergy. This is the same response, except instead of to a bacteria that can harm you, it's to an allergen like pollen or some food thing. So that's why each time you're exposed, the reaction is that much worse because your reaction gets better and better each time, right? But in this case, as far as allergy, it's not the response we necessarily want. In the terms of infection, this is the response you always want, right? The problem is our body can't distinguish between the two, right? But there's ways to trick it. We're going to talk about that later. Different therapies to try and get it to go in a different direction, right? So this is an excellent slide because it's an excellent summary of this process, right? We're going to add in some more details today, and as I said on Friday, we're going to really draw it all out together as a class and hook it all up together. So let's add in those details today. So I think we did T independent last time, right? No? We hadn't made it there yet? I know we watched this animation. I guess I should have looked at my recording from last time, huh?
Independent doesn't need a T cell to activate it.
Right, yeah. So like I said, that second step we can skip. And that's because, as we saw in those other animations, it's able to interact with a lot of the antibodies. And these are nice Ys, right? Nice, beautiful Ys with 2 antigen binding sites on their surface. So an example is lipopolysaccharides. It's kind of repetitive, so lots of epitopes of that molecule are repeated, and so therefore it's able to interact with a lot of the antibodies on the surface. Because remember, these antibodies, they only bind to a specific molecular configuration. But for this particular molecule, it repeats that configuration a lot. And that's why it's able to interact with a lot of the antibodies on the surface of the B cell. And that's enough to let it go forward, skip that step of having to interact with the helper T cell, hence independent of the T cell. So these interactions, I said, they weren't drawn in that much detail. They're very specific molecular interactions that allow these cells to talk to each other. So there are markers found on all your nucleated cells and on your antigen-presenting cells. And these are actually the markers that create a problem when we try to do tissue or organ transplants, because the immune system can't communicate with them and doesn't recognize them. So the abbreviation we use for them is MHC, which stands for Major Histocompatibility Complex. I'm going to see if I have it written out here. I know some of you guys like to write it out, and I don't. So MHC. And that's because it played a major role, histocompatibility, tissue matching, right? And so these markers were the ones that create that problem because they're not able to communicate with our immune system because they're different. So the only person who would have the exact same of these markers, these receptors, is your identical twin. So who's got an identical twin? Yeah. Not many of us, right? An identical twin is genetically identical to you, so they would make it exactly the same, right? Okay, I didn't want to cough into that. Okay, so antigens that get within our cells. So think about what gets inside of our cells. What would get into any of our cells and create a problem? What has to get into our cells in order to replicate itself? What pathogenic entity that can make us sick? Which probably got me the past couple of days. What do we worry about catching from other people? Germs, be more specific. This is microbiology. Bacteria, viruses. Which one of those is going to get inside of our cells for sure? Viruses, right? Viruses definitely have to get inside of our cells to replicate. Some bacteria get inside of our cells, but they don't have to, right? Some are intercellular parasites, others are not, right? So the most common one that gets inside of our cells would be a virus, okay? And so refer to that as an endogenous antigen, endo meaning within, okay? So all our nucleated cells, and I have this backwards, of course, right, have this receptor class 1 on the surface of our cells. So if a virus gets into your cell, it's going to pick up a piece of it and present it in the cleft of this molecule, this MHC class 2. And so it's going to present it outward so that our immune system can see what's inside of our cells. So it's like, hey, this got in, hey, right? And they're always doing this, even with self-proteins. And self-molecules are always presenting. They're always doing this. So this is a normal process for our cells, right? They're always basically showing their guts on the outside. Now, we have a set of specialized cells that one of their main jobs is antigen presentation. And this is where they actually bring stuff in from outside and process it, right? We know phagocytic cells do this, right? Macrophages and dendritic cells. In addition, the B cell does antigen presentation in order to talk to the T cells, right? It brings it in via its antibody, its B cell receptor on its surface, and then it's going to present it in this molecule. And this is MHC class 2. So anything it takes in, right, phagocytose, it's going to present it on MHC class 2. And they're doing that. They're presenting antigen. That's why that process is called that, right? They're presenting. They're saying, hey, look what I got. Look what I picked up. Now, this doesn't mean that they don't have class 1. They do. Like if a virus gets inside of them, they can present via class 1, right? Because there's a different mechanism you'll see in a moment between the communication of class 1 and class 2, right? And because it has to do with, well, what type of antigen are we dealing with? Is this something you gobbled up, or is this something that got inside of you, right? And phagocytic cells, we want them to continue eating and sharing this information, where one of our normal body cells, say a skin cell, if it's infected with a virus, what are we probably going to do to it? We're going to kill it, yeah. We don't want it to continue gobbling up more viruses, right? Or spreading viruses for that matter. Right, so antigen presentation, and I think I might actually play the audio to this.
Antigens are processed differently, depending on whether they originate within or outside the cell.
Oops.
Such as viruses or self-proteins,
That's good.
are broken down into fragments. Fragments of foreign proteins are antigens. The antigens are then transported into the rough endoplasmic reticulum. The antigens combine with class 1 MHC molecules. The combination is then transported to the Golgi apparatus and from there to the plasma membrane. Foreign antigens presented on class 1 MHC molecules stimulate cell destruction by activated T cells. Self-antigens are not recognized by the T cells and do not stimulate cell destruction. A different process occurs if the antigen originates outside the cell. Phagocytes, such as macrophages, ingest foreign particles such as viruses and bacteria by endocytosis. The foreign particles are broken down into fragments within a vesicle. The vesicle containing the foreign fragments fuses with vesicles from the Golgi apparatus containing the class II MHCs, and the two structures combine. The MHC class II antigen complex is transported to the plasma membrane. The displayed MHC class II antigen complex can stimulate other immune system cells to respond to the antigen.
So two different classes, right? Class 1 and class 2. Class 1 found on all of our cells. Class 2 is very specialized, right? Just on those antigen presenting cells, right? Not to say they don't have class 1, but mostly they're going to be using their class 2 to do antigen presentation. So once we have this information, what's going to happen, right? So T cells play a very different role than B cells, right? They never produce antibodies, right? They must have antigen presented to them, right? So some cell has to be holding it in MHC class 1 or 2 and present it to that T cell. Effector T cells directly interact with target cells, right? So they're going to communicate directly with cells and cause changes in that cell, whether kill that cell, activate that cell. So remember before we mentioned CD markers, or how we tell the difference between the two main types of T cells. So notice here, cytotoxic T cell has CD8 written right next to it. These are almost synonymous terms nowadays in immunology. When someone says CD4 or CD8, they're talking about whether it be CD8, a cytotoxic T cell, or if it's CD4, a helper T cell. Right, these are used interchangeably because just these cell types have these markers on them. And I think I gave you guys my clue for remembering them. So CD4, I remember 4 for 4H, right? So 4 is the helper T cells, and CD8, the number 8 almost sounds like eat, and cytotoxic T cells will literally eat or destroy other cells. A lot of times they're abbreviated to T with a subscript C for cytotoxic, and then H for helper. So the difference here is that CD8 cytotoxic T cells can only interact with MHC class 1. And remember, this is found on all nucleated cells, including antigen presenting cells. The difference here is what type of effector cell this cytotoxic T cell is, right? It's usually going to induce that cell to die, to undergo apoptosis. Remember, this is cell death without what very common immunological reaction? What doesn't happen when we tell ourselves to commit suicide? Apoptosis is basically self-suicide, right? But we don't get inflammation, right? That's the distinctive difference. We don't get inflammation with this. And this is endogenous antigen, stuff that got in. So the virus, this is one of our main protections against viruses that get inside. But it also could protect you against bacteria that get inside your cells, or even protozoa that get inside of our cells. Now the helper T cell is the one that helps out, you can basically think, your antigen presenting cells. And there's one that's not listed here. Right, so we said B cells, macrophages. Who's another one that's really good at antigen presentation? Starts with a D. Dendritic cells, your dendritic cells. Right, and so they all have MHC class 2. They can all communicate with the helper T cell. As it implies, the helper T cell is going to help out. And typically that's to activate whatever cell it's communicating with, whether it be the macrophage, the B cell, or the dendritic cell. With macrophages and dendritic cells, increase their killing ability and their number, right? Elicit more cells to the area. With B cells, it's going to activate them to proliferate and then differentiate into, what's our two cell types for B cells? Memory cells and plasma cells, right? Remember, secrete antibody. So for the B cell, right, we already know their B cell receptor is actually an antibody class IgM, right, out of the 5 different types. And it can interact directly with antigen. Where the B cell has just, this is the B cell. The T cell just has one receptor. The T cell receptor can only bind with just one antigen. Same thing with MHC, right? It's just one molecular complex, just one antigen binding site. So the B cell is different in that it has 2. But they're exactly the same, right? Both halves are the same. We looked at the structure last time. So this just is adding in some pictures, right? So what is CD4? It actually is next to the T cell receptor. And it's going to help that T cell recognize the MHC. So notice this is a cell. It's presenting different antigens in each one of its MHCs. When this T cell comes along, it's going to inspect this cell. If it recognizes, its T cell receptor recognizes this antigen, then it's going to communicate via cytokines as well as that binding interaction to activate that cell. But only if it recognizes it. In this case, it's probably going to cause that cell to die, right? Go under apoptosis. Because CD8, remember, is a cytotoxic T cell. CD4, on the other hand, interacting with an antigen-presenting cell, and here in this diagram they've got them all, dendritic cells, B cells, and macrophages. That CD4 is what allows it to recognize MHC class 2. Okay, so again, a little story that I tell myself, right, to remember these, right? 2 and 4, right, are multiples of each other. So MHC class 2 interacts with 4, right? And basically, again, 4 are helping out these cells. Where CD8 is going to be our primary number 1 response, right, to kill any of these cells that are virus infected or behaving badly, right? That's their number 1 task, is killing. Where number 2 is helping, right, with the CD4 helper T cells. You got to tell yourself these little stories, right, to try and help you keep these numbers and abbreviations situated. So here's an example, right? So here's a dendritic cell, right? Notice it has both MHC class 1 and MHC class 2. It's an antigen-presenting cell, so it has both, right? If it's presenting via MHC class 2, right, it can interact with a helper T cell, as you see here. Its MHC class 1 can interact with the cytotoxic T cell. It could conceivably activate both of these cells, right, if it recognizes the antigen and it communicates with them. So they'll proliferate and they'll go off and do their job, right? Cytotoxic T cells are going to attack other cells expressing MHC class 1 and this antigen. Helper T cells will interact with other antigen-presenting cells, maybe a B cell, maybe another macrophage or a dendritic cell, and help activate them, cause them to proliferate. So notice that they said that this is happening in a lymphoid organ. So remember, this is where these interactions commonly happen. These B cells and T cells and macrophages and dendritic cells exist in this area and can travel to these areas. So can you guys list some of the organs that we talked about where we have large collections of B cells and T cells that live out and wait for their dream antigen?
Your tonsils.
Your tonsils? Your thymus? No. What happens in your thymus? What happens in your thymus first? That's one of your primary lymphoid organs. One of these cells gets educated between self and non-self there. Your T cells, right? Your T cells are educating your thymus. They don't live there. They just travel there from the bone marrow, get educated, leave, and then they're going to go to places like your tonsils, right? Your lymph nodes all throughout your body. What filters your blood? Your spleen. So that one is one of the few organs you can actually live without, although I wouldn't choose it to, right? Because you have one less filter for your blood. Where else? No. You got your tonsils, all sets of your tonsils. You've got your spleen. You got all your lymph nodes. something off of your large intestines,
The gallbladder?
appendix, your appendix. And then remember all the mucus-associated lymphoid tissue, your skin-associated lymphoid tissue. So we have large collections of these cells, right, where these types of interactions will happen. And you'll get the proliferation of the cells, and then they're going to leave these areas and travel to other parts of the body. So activation of T cells, right? Dendritic cells play a very important role, as you saw in that last picture. Right? These reside in our peripheral tissues, in our skin, our mucus. They're going to gather materials by phagocytosis, and pinocytosis. They're going to bring stuff in. They have their reason why they got that name is they kind of have those long tentacles. They can actually squeeze between epithelial cells, right, in our intestines through that mucosal layer and actually grab stuff. Your reproductive tract, your respiratory tract. They're always sampling. And remember, they have additional receptors on their surface, right, that recognize different patterns, danger signals. When they detect that, they're going to grab even more stuff, right? They're like, ooh, let me gather all the information I can, right? And I almost consider them your scouts, because then they're going to travel to the secondary lymphoid organs, right? And they're going to share this information with helper T cells or cytotoxic T cells. And this is really going to depend on who they talk to, right? That whether they're displaying MHC or 1 or 2, right? Where do they get this stuff from? Remember, macrophages, when they bring stuff in, and B cells are going to present being MHC2. And then co-stimularity molecules, right? So we're talking about cytokines and different interactions that happen in the cell surface, which we're not going to get into huge detail. That'd be a whole course in itself, right? Of these cells communicating with each other. Right? They directly contact each other and send chemical signals to each other. So what's the differences that we've noticed between helper T cells and cytotoxic T cells? And what are the similarities? Well, they both have to interact with antigen that's being presented on MHC, right? Whether it be class 2 for helper T cells or class 1 for cytotoxic T cells. I like this little diagram, right? So here's a killer T cell or a cytotoxic T cell, right? So literally, what is he going to do to these cells that are infected, right? A virus-infected cell, a bacterial-infected, a cancer cell. He's going to send that signal to kill that cell, right? He's not quite going to do it like that, but, you know, it brings across the point. So this one, I'm not going to show today. So this just reiterates what we were looking at, right? MHC class 1 interacting with the cytotoxic T cell, CD8, right? And notice the cytokines are even going to communicate, right, with other cells nearby and that cell. They're going to release proteases, which will destroy the proteins of that cell, cytotoxins, and that will perforate and destroy that cell. Where with helper T cells, they're doing more of a helper role, right? So they're two are going to get presented only MHC class 2 by antigen presenting cells, so dendritic cells, B cells, macrophages. It's going to activate with cytokines. They may recognize a different epitope because remember, an antigen can be quite complex. So we have used this ability of our cells to our advantage, right? To stimulate a primary response in a safe manner. And we've even really gotten quite high tech, I guess you could say, in generating vaccines. So we've made what's referred to as a conjugated vaccine against a T-independent antigen. So Haemophilus influenzae is a bacteria, actually. And it has a capsule. And so they've taken part of this capsule, and they're not giving you the whole bacteria. They're giving you part of this capsule conjugated to another molecule so that you don't need T cell help, right? And the B cell can go right to producing antibodies against it. So the T-dependent antigen is covalently attached to the protein. And the B cell responds to the capsule, and the helper T cell protein component activates the B cell, right? So we're using two components together. One to get the T component going, and the other component to get the B cell going. Conjugated, what they mean is they put the two pieces together, right? So that we get activation across the way. So a hapten is a molecule that can bind to the B cell receptor, but it cannot elicit the B cell to produce antibodies. So this happens with people who have penicillin allergies or allergies to poison oak and poison ivy. Those small substances can bind to the antibodies, but they don't usually elicit a response because they don't get that helper signal, that co-stimularity thing. The unfortunate thing is that these things bind to other proteins in our body, and that is enough to elicit the response to where we actually produce the antibodies. Okay, yeah, and we'll leave the, well, I still have 5 minutes. Am I wrong in that?
Right.
Oh, 7 minutes according to me. Okay. So natural killer cells, we haven't talked about these lymphocytes yet. One of the major differences between them and the B cells and the T cells is they're not antigen specific. They do not have receptors on them specific for antigen. Okay? But they do have two very important distinctive helper roles. One of them we've already seen. This is a review, right? Antibody dependent, where the antibodies are actually recognizing the cell that's foreign or defective or infected. And the natural killer cell has a receptor for that portion of the antibody. Do you guys remember what we call that portion of the antibody that the natural killer cell is binding to? It's an abbreviation. FC, right? For that constant fraction, right? So it has an FC receptor. So, and then it's going to kill this cell. The other protection we have is, remember I said all your cells, and as you saw in that animation, even we're producing and putting cell proteins on the surface, right? They're always presenting MHC class 1. Some viruses, when they get inside of our cells, they shut down that system of presenting via MHC class 1. That's to their benefit, right? Because then how are we going to detect there's a virus in there? Right? We're not presenting them on our surface. But we've got a backup. He's called the natural killer cell. He comes along, and when your cells aren't displaying MHC class 1, that's not normal functionality for that cell. Something's wrong with that cell. So what is he going to do to that cell? He's going to destroy it, right? Just as his name says, naturally killers, right? They're going to destroy that cell. So the two things, right, is any cell not displaying MHC class 1, not displaying it at all, it's going to eliminate, right? And as I said, some viruses actually will cause our cells to shut down that mechanism. Well, they won't last too long. The natural killer will hopefully find them. And then the other way is if you have antibodies to a specific antigen, right, that may be displayed on the surface of a cell, the natural killer cell can bind to this antibody and kill that cell. But there's only one class of antibody we produce. It's actually not the most abundant produced, but the most abundant in our body. It's IG. This one can cross the placenta too. That's a big clue. IgG. There you go. You guys have been studying some, right? So this is just written out in words, right? What I just said on our pictures. So again, that FC portion of IgG antibodies is what the natural killer attaches to, right, and causes that antibody-dependent cellular cytotoxicity. Or if they're not displaying MHC class 1, which is really important in some viral infections, it will kill that cell. So next time, we're going to talk about how antibodies are made different, and T cells, for that matter, receptors. And then Friday, we're going to draw on the board, right? We're going to get up, and we're going to be active on Friday. And you get 10 points for being active on Friday, so you definitely want to be here.